And first of all, I should like to express my gratitude to Professor Ivanise for this invitation. And I will talk about electrophoric and oxidative stress in cancer progression. First of all, oops, what's the matter? It doesn't advance my slides, or maybe like this. Yes, okay. So first of all, I should like to mention that the cancer is a complex disease, and we can distinguish hallmarks of the cancer. So it means the features, specific features, which highlight the development of cancer. Among these features, we can find different processes like autosufficiency in growth, insensitivity to anti-growth tissue invasion and metastasis, unlimited replication potential, and finally also voiding of cell death. And of course, different strategies have been developed during evolution for creation of these diseases. They are biological processes, but also we know that there is environmental action which should be taken into account when we talk about the cancer. So here you can see also one thing which seems to be important in fighting against cancer. This is the development of EGFR inhibitors. And my talk will be around this receptor, which is important for many processes. And that's why I highlight it here. So what kind of environmental factors are involved in the development of cancers? First of all, these are our lifestyle, and the diet, different environmental pollutions, and so on. They can cause oxidative stress, and which is an imbalance between antioxidants and free radicals. What mean free radicals? These are oxygen or nitrogen derivatives which contain electron and external electron, and which can reactivate these molecules. Here you can see that this replacement of the electrons can be important in the development of reaction change in the human body, and it can bring to the development of cancer. Here, just to remind you what kind of radicals we have, because many radicals are derivatives of oxygen. Oxygen itself, it's a potential B radical molecule, as it mentioned here, and it can undergo to the different modification, because the external orbit of oxygen can very easily lose the electrons. There are a couple of electrons which can be introduced into different reaction. First of all, when the electron the electron is lost, it can form the first free radical, so-called reactive oxygen species. This is superoxide. Superoxide is very active molecule, and it with very short lifetime, it can react with many products without any enzymatic activity. So when it accepts the second electron, the molecule becomes more stable one, so-called hydrogen peroxide. Hydrogen peroxide, even stability of this molecule allows to use it as an antibacterial agent. You can buy this agent in the pharmacy. But at high concentration, hydrogen peroxide also becomes reactive because it can be transformed, as it mentioned here, to the hydroxyl radical. Hydroxyl radical, it's also very important reactive oxygen species, and also with very short half-life. So it's less than microseconds, as in the case of superoxide, and also it can be reacted with many different biological sources. And finally, this reaction with this hydroxyl radical leads to the formation of the water. So again, to remind you that oxygen itself, it's a B radical, it is still stable. However, at some condition, it becomes very reactive by transforming to superoxide. Superoxide, again, when it takes the second electron or lost its second electron, it can be transformed into peroxide by formation of the second radical. These two molecules are agents, free radicals, which cause usually the oxidative stress. And in the cells also, there are antioxidants, which exist already in the human cells, or they can be created artificially by chemists, by biologists. And what they do, this molecule donates the electron to free radical, and this allows them to inactivate the action of free radicals. So here we have a consequence of this of the action of free radical, this air pollution, which represents a very important health problem. And we can observe the formation of aerosol, organic aerosol, for examples. Among these organic aerosols, they are so-called secondary organic aerosols. And studying of these aerosols became not so much time ago. And what are these secondary organic aerosols? These are small particles of the size of less than 2.5 millimeters, and which can be present in the air, and we are almost in a permanent contact with this aerosol because we use them, and they can be a reason of the development of oxidative stress. So oxidative stress, again, I remind you, this imbalance between this normal situation and the antioxidant actions in the human body. Oxidative stress itself is responsible for the development of different pathological processes, like it can go through the inflammation, apoptosis, autophagy, and altered immunity, or in another direction. In any case, it becomes a problem for development different kind of diseases, including cancer, Alzheimer's disease, Parkinson, diabetes, and so on. Here now we have a general pictures of this action. So as you can see, that free radicals, which can act on the level of cells, and the sources, intracellular sources of these radicals are different. It can be related with the activity of the mitochondria, or it can be peroxisome, or it can be related during hypoxia, for example, with the so-called Barber effect, and also nucleotide adenine diphosphate oxidase is located usually in the cytoplasm, and also other structures in the cells. So the formation of reactive oxygen species, as it mentioned here, is responsible for the development of different processes. These processes, as apoptosis, autophagy, and even the creation of the drug resistance, DNA damage, tumor progression, all of this can bring to the development of the cancer against more. Here, now we have a list of different receptor tyrosine kinases. Tyrosine kinases are located in the cytoplasm, and all of these tyrosine kinases, there are 58 tyrosine kinases. So they can be grouped according to their similarity. And here is the first one. We have 4 proteins, 4 receptors, epidermal growth factor receptors, 1, 2, 3, 4. And the EGFR, which was the first one discovered among all of these receptor tyrosine kinases, with a Nobel Prize obtained by Stanley Cohen for this discovery, plays important role in the development of the cancer. So I should like also to mention, because it can be used in my talk, that EGFR also is able to go to make a heterodimerization, at least with this group, insulin-related receptor. And this heterodimerization indicates that the activity related with the EGFR can be transferred to the insulin receptors. Similar situation exists also between some other receptors. Here we have a structure. We have a structure of EGFR. Normally, it's a monomeric protein, as you can see, which has 2 parts located, one in the extracellular region and the second one in the cytoplasmic region. And also there's a single alpha helix which goes through the membrane, cytoplasmic membrane. The activation of this receptor depends on the cognate ligand. So this is the epidermal growth factor itself, small protein, which binds to the monomeric form and transforms it to the dimeric form. You can see here this beautiful structure which goes through the topological change and forming the binds to the second molecules, and we have these dimers. The dimerization is very important because, first of all, this dimerization is necessary for the activation of the receptor. So it seems to be that there is some kind of topological action when this change leads to the formation of this dimeric form and the activation of the ATP binding site. ATP binding site, so itself triggers the formation of phosphorylated amino acids, among which the most important are the class of tyrosines, the different amino acids, among which tyrosines plays important role. And tyrosine phosphorylation, it's practically the beginning of the triggering signaling pathway, as it mentioned here. You see dimerization, and this is the beginning of the signaling pathway in the cells, downstream signaling pathway downstream of EGFR, so-called RAS-MAR map signaling pathway, PE3K, AKT, JAK-STAT pathways. There are also some pathways in which participates EGFR. And what we have at the end, we have very important function which have been affected by these signaling pathways. It's a proliferation, motility, cell survival, cell cycle, and so on. Many functions which can be affected by the phosphorylation. Now, to study EGFR, because we were very much interested in the development of anti-EGFR therapeutic small molecules, we decided, first of all, to subclone extracellular regions, supposing that it is the most important part of the EGFR, subcloned it, purified, and to label with fluorescent dyes, and to use for screening of libraries of chemical libraries which were available at that time. So, and we use, for example, National Cancer Institute compound libraries, about 3,000 pharmacophores. So it means that preliminarily selected molecules which possess some kind of biomedical interest, and try to find the molecules which can be interesting for us. So again, I should like to repeat that we were interested, first of all, in selection of inhibitors of the epidermal growth factor. Here, just it is not related with my talk, but it's a beautiful picture which shows the development technology, so-called small molecule arrays. And you can see here to the left this porphyrin, different kind of porphyrins which have been spotted on quadruplet. And after that, you use the spotted molecules for binding to the protein which is interesting for you. In this case, it was a fluorescence labeled Hellman serum albumin. And it was very important for the selection of the best porphyrin derivatives for photodynamic therapy of tumor. Here, the real process which takes place. So as you can see here, you have spotted molecules. As I mentioned, in one glass slide for microscopy, small glass slide, you can spot about 3,000 or even more, 3,000 and more molecules of your interest. They have been spotted, and you can use the protein of your interest. There are two possibilities to choose the molecule which is interesting for you. First of all, if there is a binding of the protein, labeled protein, you will have a signal which appears here. Probably it is, for example, some kind of control which can be used for in this binding assay. And after that, you use the second binding, or you can add to find out whether there is another molecule which can bind to some of these small molecules. Here you have, for example, that there is not competition between them, or it was another binding, another molecule which can bind to the protein. So this technology is high throughput technology, which can help in advancing your projects, and it's straightforward. And indeed, it was important in our case. And we discovered a list of about 15 different molecules which can bind to different proteins, including also EGFR. And with one of the molecules took our attention. It was in this case here. As you can see, you observe binding to the EGFR among other proteins. So it was very interesting because, again, here the proteins have been, we used them binding to the labeled molecules. And this compound have been selected. It's a so-called nitrobenzoxidazole or benzofurazane molecule. I will call more shortly NBD molecule, which help us to answer many questions. Well, now, first of all, as I told that EGFR should be dimerized by epidermal growth factor. Here we can see that such a property also has this MBD compound. It's more important to stimulate this dimerization as compared with a HER2 protein. So it seems to be there is some specific action related with the extracellular region of the protein. We suppose that probably there is such kind of cysteine or histidine which could be important for this interaction. So the second thing which we observed this activation of the phosphorylation pathways. As you can see, that there is a tyrosine phosphorylation when you use the compound here, or another compound also, which will be described a little later. And we have also phosphorylation of downstream signaling proteins, like here CHC, ERK2, CBL, GAP, and this is tubulin, which can be used as a control. So the compound was able to provoke or to stimulate the dimerization of the EGFR and also to trigger the the signaling pathway in downstream of that. Here you can the action of the inhibitor, which shows that when you use inhibitor of the EGFR, it can block the phosphorylation or diminish the phosphorylation of EGFR. Here we applied another technique that can be also very useful. It's a so-called antibody microarrays. So it means that 50 different receptor tyrosine kinases have been antibodies have been spotted on the glass slide. In this case, it was a nitrocellulose surface, and using the binding assay. In the first slide, we have a binding to the compound which was to MBD, which was discovered. You have seen here, first of all, binding to EGFR, and also to some other protein, including here, for example, binding, very good binding also to the insulin activation, maybe it's better to say activation to the insulin protein. If you compare this binding, which has been done also with EGF in parallel, we have a similar picture for receptor tyrosine kinase. But as you can see, there is not activation of some other receptor tyrosine kinases by EGF, whereas there is a clear lower, but anyway, binding to or activation of the receptor tyrosine kinases by embedded molecule. Here we have an explanation. I don't present similar results obtained with in other experiments. We have indeed clear explanations that, first of all, the compound, embedded compound, is able to activate almost all three signaling pathways which have been studied in our research. But what is also important, it can also activate the nuclear EGFR, which is responsible for the expression of the some protein like cyclin D1, ENOS, MIB, and so it means that the molecule behaves as a real activator of EGFR, which seems to be similar to the action of EGF itself. However, here we can distinguish some differences which were characteristic for this molecule. First of all, similarity, we have observed that at least two tyrosine residues in EGFR can be phosphorylated by EGF and MBD compounds. We have activation, which was studied in details, of MAP and ERK cascade signaling pathway. We have also activation of ubiquination degradation cascade, which is responsible for the degradation of the protein, and transactivation between EGFR and RB2 and RBB3 and insulin R and insulin growth factor 1. So through the heterozymation. And finally, the molecule was active in different tumor cells in breast cancer cells and also in lung cancer. But there is also some differences which was very important. So induction action requires high concentration, and the compared to the action of EGFR, which activate the EGFR in 1 minute. So we this MBD requires longer time, more about 5 minutes, and it was also active in serum starved and non-starved cells, what was not the case with EGF. And also we can see here transactivation of ROR1, EPH, and MER. And finally, the I can highlight that it seems that the compound was able to go through the rapidly through the cytoplasmic membrane. We decided to enlarge the list of MBD compounds and to make comparison to understand better how these compounds can activate the EGFR. So here, new compounds which have been found, detected, and used in our work, which came from the Paris scientists in Paris. And here, for example, one of the experiments, which also was very important, that this compound can activate EGFR, as I mentioned, but at the same time, they can dephosphorylate another protein, which was supposed maybe this activation is related with the loss of the with the activation of phosphatase, which removes usually phosphate groups, so-called PTP1B. EGFR itself, it's a substrate for PTP1B, and this experiment clearly showed that the compound has two different kinds of action. First of all, these opposite actions are related with the phosphorylation of EGFR from one side, but the second one, this dephosphorylation of PTP1B. It was very important for us, which allowed us to suggest that probably this compound is responsible for the formation of reactive oxygen species. And we started another step of our research to understand the behavior of the molecules. Here, there was already explanation of the activation mechanism of PTP1B by hydrogen peroxide. Here we can see that in the case of PTP1, we observed the formation of sulfonic acid in the structure of this phosphatase. This is cysteine 215, which is transformed into sulfonic acid by forming another derivatives. So in the case of EGFR, there is also generation of H2, which can be promoted by binding to EGFR and followed by the production of this reactive oxygen species by NADPASH oxidase. So it was important suggestion which helped us to explain what we observed in our case. What is also important, we find out that all compounds are fluorescent. It was a really key point to understand the mechanism of the action of these molecules. Here, these compounds, as you can see, possess different log per value. Log per value, this important therapeutic parameter which can help in the prediction of the transfer of molecule through the cytoplasmic membranes. They can be positive, more than 0, It means that they are lipophilic, such kind of molecules, or they can be less than 0. In this case, they cannot be transferred through the membrane. So it means, again, this log-per value, important point to understand correctly the behavior, predict correctly the behavior of the compound. Indeed, it is the case. Here you can see that these two molecules, the 55, I will just use last figures here. 55 and 43 they are positive and they can go through the membrane, whereas others, or MBD, even if it is positive, but it is another effect, it damages strongly the cells and it cannot be used in analysis. We can observe that they can go through the cytoplasmic membrane. It was important consequence in explanation of different studies, different results obtained in the next steps. Here, what's the matter? Okay, so first of all, to confirm that this lipophilicity is important for the transferring of the molecules. As you can see, the compound 55 can go through the membrane, and here you can, in a green color, we have a presence of this compound in the cytoplasm. Red, these are membrane, both sides of the membrane, if you want to look here, and blue one, it's a nucleus, which also has another color. So it is not the case with, for example, non-lipophilic compounds, because practically no molecule in the cytoplasm. Or in the case, as I mentioned, MBD itself, it's damaging, it's destroying completely the cells and doesn't allow to follow the action of the compound. So lipophilic MBD compounds rapidly, in 5 minutes, are able to go through the membrane. And of course, they can make something which is important from our point of view. Indeed, we use special techniques to develop the formation of reactive oxygen species. You can see here, so it's the case of HD2, and in red color, we used PEG catalase, because PEG catalase could block the it can be used to prevent the further action of the compound. And here we have a different kind, but anyway, different types of the formation of this peroxide hydrogen here, as in the case of the action of EGF. So we have a proof, direct proof, that hydrogen peroxide is very important to the formation of the reactive oxygen species. Here's the same picture. We can see here there is nothing, but slowly there is appearance of the signal, of the fluorescent signals that highlight that the molecules is active in the cells. Next one. Here, in this important picture, we observed the the action of the compound. And here you can see, first of all, to the left, this binding of the MBD compounds to one of the two, maybe the most important enzyme, third one, superoxide dismutase. It's in a eukaryotic system, it's coenzyme dependent enzyme. So it's a 15 kilodalton protein. It's about here, you have 15, 16 kilodalton protein. So, and what we have here, we have an action of these compounds on the formation of dimers. You see, the action of 155 and 43V leads to the formation of dimers and 32 kilodalton dimers. It is important to mention that these dimers are rather stable. They are stable to a low concentration of superoxide, SDS. And it was interesting and surprising for us to find out such kind of action. And in this picture, we can see also when we use the anti-reactive oxygen species, so like in the case of the N-acetylcysteine and thioglycerol, so that we can observe that there is no practically the formation of bonds to the superoxide dismutase. So it's a position of 16 kilodalton in our case. This is a control, positive control. This is just an inhibitor of EGFR. And you can see in the case of the classical inhibitor of the EGFR, there is a still binding to the EGFR. So superoxide dismutase exists in this case. Sorry, go back again. And to the right, we have another picture, which is important. So here is a chromacy coloration in the classical juice. We observed also, we can observe that there is a monomeric superoxide dismutase, and the compound, in this case, it was a purified, of course, okay, because as the case here. So, and we can observe that all of the compound, lipophilic and non-lipophilic, are able to promote the dimerization of NBD. And what we observed also, that it can be observed using fluorescence detection. Fluorescence detection has been done in particular way, and I will mention it just a little later. So, go back. We suggested that if antioxidant prevent SOD1 dimerization, so it means that embedded compounds preferentially bind to SOD1, and SOD1 could be involved in the processes followed after such kind of binding. We tested also the action of these molecules by inactivation of SOD1 using RNA system, RNA inactivation system for SOD1. We performed it in knockdown cells, and we can see here interesting situation. In knockdown cells, no action practically. SOD1 is inactivated, whereas in control system, after 5 minutes or even 15 minutes, we observe again the appearance of dimerization forms in the action, by the action of these compounds. First of all, here it's most important by 155, and less with another one, 43V molecules. So we can observe the phosphorylation, which is related with the formation of this, on the case of the control experiments, with the action of the embedded compounds. Next here. So this is the activation mechanism which can be summarized of EGFR. I will not go deeply to the understanding because it's not related directly with our talk, but just I wanted to mention that here we have again the formation of H2O2, which goes through the NOx2 mediated transporters in the cell, and whereas in our case we have another action, another type of action, it is related with the formation of, with the binding of the compound to SOD1, which results in the generation of hydrogen peroxide. So, and we found also that the EGFR has can be activated by two ways: through the action of the compounds on the extracellular region, but also through the action of the compounds on the cytoplasmic region. It was a cysteine 797 residue, which is very close to the catalytic site and usually is responsible for the activation also by OGF. Here, now we have another picture, which is also very important. So we used to show that compounds are able to bind to different proteins because we have a preliminary results indicated on that. But it cannot be done in the gel itself because of different action related with the gel inactivation of the fluorescence, such kind of extinction coefficients, quantum yields, emission spectrum, and so on. To avoid this problem, we transferred the proteins from gels on the membrane. It is not Western blot. It's simply we use a similar approach which has been suggested for Western blotting. Transfer the proteins to the membrane and to use a special detection system, fluorescence system. It's a Python system which can detect the signals at the corresponding values required for the detection. So it was an excitation at 480 nanometers and excitation, sorry, and with the emission at 520 nanometers. And we have a similar picture for 4 different types of MBD compounds. I should again to repeat that here we have a results of the action on the proteins which have been extracted from the cells. So proteins are available for the action. And we have such a picture which appears in 5 minutes already. Here, when we use now the action on the cells, we have a different kind of pictures. This electrophoresis of living cells, and we have no difference between the action of compounds. However, a great difference we observe here when we make fluorescence of the proteins which theoretically could bind to the MBD compounds. Only lipophilic treatment, a treatment with lipophilic MBD compounds allows the detection of the proteins. No detection of the proteins in the during the action of non-lipophilic molecules. The different proteins starting from 15 kilodalton to about 400 kilodaltons appear during the action of these lipophilic embedded compounds. The intensity of binding of bound proteins is different. They are very strong binding, they are also weaker one. At least we have probably 100 or even 200 proteins which have been bound to the MBD compound. So it is very important because it was an origin of the explanation of the mechanism which is involved. Here, another experiments to show when it starts, and we can see that first of all, it requires more than 1 millimolar of the compound. So probably even it works at 1 millimolar, but it is the sensitivity of the technique which have been used. And we can see starting about maybe between 4 and 6 minutes, maybe about 5 minutes, we observe the appearance of these perfect bands. And these bands are still visible up to 10 millimoles of the non-embodied compounds which have been used. So we have a very interesting situation when the compounds start or provoke the action after a few minutes of the introduction into the cells. What is the matter? What happens? How can we explain this mechanism? So fortunately, it was already described that the compounds undergo to the chemical modifications in the cells. Here, the compound, one of the compound of the MBD derivatives, which has been described chemically by very precise in experiments, very precise experiments. It is a very potent molecule, very potent generator of peroxide hydrogen. And here we can show what happens during the action of these molecules. So it first of all reacts very quickly. It's chemical. These experiments have been done not in the cells, okay? They have been done, first of all, externally, and after that, they have been confirmed in the cells. We have a very interesting situation that there is, again, loss or donation of the electrons, and with the formation of the electrophilic molecule. Here is a nitrogen residue. We have this point, this indicates that, okay, it becomes only electrophiles. But electrophile molecules, as I mentioned, they cannot exist long time. They should find another compound for joining, and they, because of that, they disappear. And what happens in this case? Normally, it can be in the cells can be different proteins to which they bond. But in this case of this potent agent, they was observed there is no possibility to continue the formation of donation of the electrons to another protein, and they go back to the hydrogen peroxide, and they form so-called cyclic pathway. So it means that cyclization of the formation of this, okay, of the peroxide hydrogen, and the cyclization which called redox cycling. So it's very interesting situation when this redox cycling is formed, and it doesn't need, or even it cannot continue its action because it finds already the recipient for forming, for inactivation, but or activation, because both are related, and we have a cycling of this situation. But the question rises, just a minute, if there is a recyclization of this electron, what happens with another part of the compound? And this is the explanation. So here we have only the reactivation of the oxygen in this redox cycling. So it means this compound is left alone and it should react. That explains the appearance of many proteins after the binding to this embodied compound. So it means that it can provoke so-called electrophilic species which are similarly reactive, or less, of course, but anyway, they are able to also bind to the protein. And we have, indeed, as I mentioned, binding to multiple proteins, and it leads to the formation or to the generation of the electrophoric stress. So here to the right, so another thing, they bond to SOD1, and to explain how it happens. And this redox cycling goes through the SOD1 dimerization. Here we can see that the compounds goes, okay, back and to, again, binding to the precursor and provokes the formation of the cycling. So it means that excess of peroxide hydrogen or hydrogen peroxide is responsible for the formation of this redox cycling. So at the same time, it can bring to the oxidative stress two processes, electrophilic stress, oxidative stress, which function together. This simultaneous formation of two different species, well-known process of the formation of oxygen species, and the less known process, the formation of reactive electrophiles, or I will tell reactive electrophilic species, RES in abbreviation. So we have now explanation of these processes which has not been described up to the time. Now, let's make some conclusion, what we have observed. First of all, that the gravity of the electrophilic stress depends on the aberrant state of certain dimers, which provide toxic concentration of reactive oxygen species, and when cultivated with inhibited compounds. So two processes, two different processes, but inseparable processes because they start from the same molecules. This is the electrophilic stress and oxidative stress, which apparently reflect the transition of cells from normal stasis to hypostasis. Hypostasis because cancer usually is related with such kind of oxidative stress, with hypostasis because of the increase of the hypoxia in tumor environment. And what is important for definition of the electrophilic stick, that I can tell that overwhelming production of reactive electrophilic and oxygen species that excess the ability of anti-chemoprotective and antioxidant system to neutralize both types of reactive species. At the moment, it is impossible to distinguish directly whether there is electrophilic stress or oxygen species. However, we can suggest that in some cases, the dimerization or formation of stable dimers of superoxide dismutase can help to make this distinguishing, because as I mentioned, we observed the formation of superoxide dismutase, stable dimers of superoxide dismutase during the action of our embedded compounds. Here, a general view on the processes. So it can be explained by the, here, sorry, can you go back? So by the action of the embedded compound, which should go through the membrane. If they are non-lipophilic, so they cannot go through the membrane. And if they are lipophilic, they become reactive substances. And they go entering in the cells, provoke the modification or transition of the oxygen to superoxide, this superoxide oxygen, and here it leads to the formation of the redox cycling. So in redox cycling requires also the binding to the superoxide dismutase. This superoxide dismutase so can be important in the for the binding to embedded compounds, also to the different cytoplasmic proteins, or even in nuclear proteins, as we have shown for DNA protein kinase, and also for other cytoplasmic proteins, again here, like phosphatase, or even for binding to the mitochondrial proteins. So we have an common action of two different processes, oxidative and electrophoric stress, which are important as a consequence of the action of this molecule. Of course, the final result of this action is a progression of the cancer. And here, the last picture regarding my talk, the embodied electrophiles and reactive oxygen species behave as independent entities. These entities, the groups, are indeed different, and because they have a different recipients, proteins, or even they make a competition for the binding to the same proteins. Of course, the binding site for these compounds should be nucleophilic amino acids in the proteins. So, and what is interesting also, that accumulation of peroxide hydrogen and concomitant embedded electrophiles is a cause of oxidative and electrophoric stress in the cells. It raises the question about the necessity to be very careful with the pollution. So we suggest that it can be done by using microarray-based method to assess the pollutant exposure. But it's interesting question for the future scientists, for scientists who work with the pollution. So identification of biomarkers of electrophilic stress is a prerequisite to develop alternative strategies in fighting cancer and neurodegenerative diseases. I should like especially to mention this because there was attempts to use embodied compounds as an antioxidant, and at least four important studies in different countries stopped do that because our data indicated that the formation of this reactive electrophilic species leads to the binding to different proteins. Of course, inactivation of the protein function can be important and can eliminate the positive expected action of these molecules. And that's why, so my feeling is that it is impossible to construct small molecules which can completely block the action of this reactive oxygen species because of the concomitant transformation of this electrophilic species. Therefore, if you look carefully at the literature, you cannot find a good molecules which have been suggested during many years. There are some action related with the diminution of the oxidative stress, but there is no strong evidence that they are indeed very useful in the fighting against the oxidative stress. So oxidative stress is a big problem and requires more attention for studying to suggest alternative ways for the development of antioxidative molecules in the future. Here, it's a pleasure for me to suggest these technologies, microarray-based technologies, which have been used with small molecules which can be spotted and, as I mentioned, used for the detection of the activity of these molecules, and also antibody and protein arrays, which can be used also for the following the functions of these molecules. So these two technologies are straightforward, high throughput, multiplex, highly sensitive, rapid, and cost effective. So practically, these technologies can be used from starting from the selection and screening of libraries, compound libraries or protein libraries, and go up to the preclinical studies. I suggest if you are interested in these technologies, you can join me, you can write an email, and I will give you some information, and even I will tell you that practically you can use these technologies without any limitations. So my feeling is that already I have finished, and here I should like just to thank my colleagues who participated in this work from University of Nantes, from University of Paris-Descartes, and also from Institute of Fine Organic Chemistry in Yerevan, and also from a small company, Prodnetomics. The work has been completed somewhere in 2018, but now we restarted to make some job in this field because the problem is very interesting and very important for the future. So thank you very much. With a pleasure, I will answer your questions. If you have questions, please. It seems you have.
Please, your question. Thank you, Professor, very much for this interesting lecture. Does anybody have questions, please?
I can listen.
Oh, I think your lecture was so clear, and nobody have a question.
You have no question, okay.
So I'd like to say thank you for joining us in the frame of summer school. It was very interesting for us, your actual information, and I hope that maybe somebody of participants want to join you to know something.
Okay.
Thank you very much.
Thank you.
And now we have a 20 minutes break before the second lecture from Olena Żypiszewska. See you later.
See you later.